because of the type of fields that I deal with. I mean, you'll be seeing some of that today. I'm here to talk to your Biology 427 class about what is environmental DNA, or eDNA. The long story short is that if you run your finger across the desk ahead of you, that's in front of you, or just touch any surface, and you were to extract that DNA that was attached to your finger somehow and see what's there, that would basically be environmental DNA. And so that's sort of the concept I'm going to talk about today. I'm going to go through the history of where environmental DNA came from as far as the idea is concerned and how we got to where we are today, as well as the development of the different methods and the problems that we ran into, and my modest contribution to some of that through my graduate work. And then we'll talk about a little bit about the lexicon of what we have now in terms of environmental DNA, as well as some of the work that we're participating in at Longwood University nowadays with freshwater mussels. If you go back through the history of environmental DNA, depending on who you ask, there's competing narratives, but I tend to lean more towards the Eske Willerslev side of things. There's one other group that started working on bacteria from the environment in a similar time period, but it was for a different purpose, and it didn't really lead to the sort of explosion of methods that we're talking about. So I lean towards Eske Willerslev's beginning here from 1999 because his curiosity was really based in whether we could learn anything from DNA that was in the environment, since our sequencing methods were getting so much better. This guy I highly recommend looking into because he's just had a really prolific scientific career in a way that we don't see very often anymore. I'm going to kind of describe him as more of a naturalist, and as far as like you think of people like Charles Darwin is concerned, he's really interested in getting into the world and trying to find things that are in the world that other people aren't. Him and his brother have done a lot of research that's been fairly influential over the years. Eske a little bit more than, I forget his brother's name now actually for that reason, but they're both prolific scientists, and both of them were originally thinking about the environment and what sort of things they could learn from what's there that we can't normally see, especially in the context of sequencing. Their interest was really sort of in deeper time questions about what sort of communities were present in at least the bacterial communities in like ice cores, or what did the world look like from a microbiological perspective like 10,000 years ago in ice? Could we learn anything about that to recapitulate the ecosystems? And in fact, what they were really interested in was learning like a visual idea of what the world looked like, maybe at the last glacial maximum, for example. And so that was really the genesis of this idea of eDNA that you can see a figure from here on the right in the slide. This is from that publication in 1999. They were doing some Sanger sequencing to understand what the bacterial community was like in ice cores at that time. And I put this figure up here in this case just to show how different the inputs are from 1999 to now. This is Sanger sequencing data that in our modern parlance would be pretty bad, but I think for that time looks pretty good, especially considering that they put this together as a figure within their paper to get a consensus sequence, which you can see at the top of each of these. So it was a lot of work at that time to reconstitute what the sort of 10,000-year-old ice core world would look like for eDNA, but this was the initial idea. He goes on to do a ton of things with regards to ancient or environmental DNA. He kind of heads down the ancient DNA realm a little bit more, but you could argue that ancient DNA is kind of a subset of environmental DNA, but all of this work is really important because of what I'll show you in a little bit in terms of other people picking up on the idea and really asking more questions of what we could do with DNA that's present in the environment in various forms. But he goes on to do a bunch of different things, like he describes the first ancient human DNA at all. He describes the power of population structure from ancient DNA in humans. I think that's the one where he starts describing population structure with Neanderthal DNA, but then additionally, you probably heard of the, at some point, of the sequencing of the first complete genome of ancient humans, in this case, Neanderthals from permafrost, the preserved hair. He did all of these really big things, and we were able to look at sort of historical distributions of genetic diversity and how that interacts at that sort of level as a result of him. And all of these pieces of work promoted an idea that DNA is pretty much everywhere, but from every taxonomic level, from bacteria to humans, we can get DNA from old sources and surfaces and matrices that we didn't think was possible previously. I mean, In this case, we're looking at ancient human DNA from permafrost preserved hair to be able to get the first complete in quotes, genome sequence of a Neanderthal, right? So it's pretty amazing. And this is the kind of concept that we're talking about with environmental DNA. These ideas, this sort of idea from him sort of led to these other different questions that were kind of independent with, I wouldn't say directionless, but sort of took the ideas of like looking at ice cores and was like, well, if we can do ice cores, maybe we can track things in water. So this person in Spain tracked frogs initially in water in 2007. Simultaneously in the United States, Karen Goldberg tracked frogs and salamanders in a similar capacity, and that led to a publication in 2011. It also led us to start thinking about the problems if we were going to use this as a monitoring tool through the gene, which is also in 2011, looking at the persistence of eDNA in water. So these two first ones were basically like, can we see environmental DNA in the matrix of water at all for any species? And the answer is yes, to some degree. And this is all presence-absence data. The next sort of thing here with the gene is that, if environmental DNA is in the environment, a la, like I mentioned, you can take your finger and run it across the desk in front of you and you'll probably pick up some eDNA in your finger. How long does that maintain itself in a way that we can actually, assay it back up and be able to tell what's there? That's the question that they were trying to answer with this. And then Thompson and some others looked at several freshwater species, sort of like a barcode, like the beginnings of barcodes that came out in 2011. But at this time we had ideas and data that began to trickle out that was kind of directionless, if you will, at least from a large perspective. These are huge steps forward in as far as we're expanding the number of matrices that we can get environmental DNA from. I mean, we started with ice cores, we went to ancient human bone and hair, and then we went to water, and then we sort of stuck in water for a little while, but there's multiple matrices that can be, eDNA can be isolated from. And this really sort of was summarized by this review paper from Thompson and Willerslev himself that wrote a review that summarizes the work in 2015. And I would say this figure on the right is probably the most sort of prolific and influential figure for environmental DNA in the field. What's really showing is sort of what I was articulating. So it's giving an idea to everyone, like the base methods of like how you could collect eDNA from anything, right? So like, and more formalizing the methods such that instead of somebody going out and just sort of independently gathering materials from some matrix and like, look, I found this salamander here and I got this arbitrary volume of water there and it showed me some frogs. No, it's like this is starting to articulate the sorts of methods that you could use to be consistent, to be able to expect consistent return of eDNA from whatever matrix you're working in, be that, they're showing here like water, or in this case, ice cores, water, soil, various like plant lives. There's all these different matrices that you can pull from and then get into, some sort of DNA amplification of some sort, PCR, qPCR, Sanger sequencing, next generation sequencing. You put it through whatever device you need to, and then you get the sequence back, and then you understand the presence of the sorts of animals or plants that are present in either now or in, further time. So this is a really big paper, and as far as it starts to begin to put together the methods in a way that isn't so disparate, because the if I go back one slide, these were all just kind of coming out independently, and it was really cool to see that we were able to track frogs and salamanders and look at the persistence of DNA in water, and tracking a bunch of freshwater like fish species for the most part was what Thompson was working on. But then when this review paper came out, it really sort of summarized all those things in a way that hadn't been done yet by 2015, right? So, and actually this was right when I was graduating from grad school, so that's why my modest contribution, it's not cited in this, but like all of this stuff was kind of happening at the same time that I was performing my graduate research. So I sort of missed out on this summary, because I think this came out right around the time that I was putting together my sort of thesis document to be able to go and defend my graduate thesis. By the way, I think I failed to mention this earlier, but if you, I'll give the slides to Dr. Beach, And all of these articles are hyperlinked in, so you should be able to go and get these papers and take a look at them if they pique your interest. So after we have this sort of big review paper, this is from a similar one at the same time called Conservation in a Cup. It came out in 2012. Not as prolific as far as summarizing eDNA, but I like this image because it shows sort of the things that have been gathered for eDNA at the time between like salamanders, frogs, insects, aquatic macroinvertebrates, fish, and aquatic mammals. Those two things kind of gave us this working definition of what environmental DNA is, and it's extracted and amplified from water samples, and the amplicon is used as a surrogate for presence. And even to this day, the environmental DNA, for the most part, has been developed to monitor and learn about behavior of species in the context of rare and endangered, indicator, and invasive species. Now, I will separate sort of bacterial source tracking a little bit from environmental DNA, because you could put looking at the microbial community under environmental DNA, but at this point I would say they're a little bit separated, although the methods are super similar as far as how you would use the amplicon in some form, that'll be used as a surrogate for presence. Either you're using it for bacteria or you're using it to track mammals, but the difference is the complexity and the ease by which you can get these things in, because the matrices have all these different sensitivities and different methodological differences that can be customized even for every individual research thing, which is why it's important that previous review paper from Thompson and Willerslev came out, because they really sort of started to unify the ideas around the methods. You would still have to kind of tweak the individual methods for whatever project you're working on, and I'm going to talk about that in just a second. But at least at that point, we had an idea of how you could collect something from one of these matrices and be able to use the amplicon of whatever you extract, from the extracted DNA, to be able to use as a surrogate of presence in some unified form at this point. So this leads to an explosion of methods questions, because we sort of summarized those methods, came down on a unified front, which is really cool. It came out to, like, instead of people just identifying that there's some species present somewhere, it's like, okay, we can do that now, but what are all the limiting factors for the methods that are occurring? And it led to a lot. I mean, all of these are once again linked. So we talked about previously, like the persistence of eDNA. That kind of was like randomly landing on this, but all of these methods papers that I've highlighted here, of which this is just a subsample, there's a bunch from this time period. They're all from like the 2012 to 2017-18 time period, where people in Japan with Takahara, for example, started using biomass as a proxy for understanding how we could use eDNA to actually quantify. So instead of just having presence-absence, whether something is there or not, actually having a degree to which something is present. So there's a lot of methodological questions about how you build up a biomass curve to understand when you're using qPCR, for example, what CT value is equated to what biomass. There's a lot of work to go into that to be able to model it. And this is one of the first attempts here from Takahara with some carp species in Japan. There was another paper as a result of all these methodological questions that talked about, like, okay, well, we're getting this eDNA in the environment. We have some idea of the persistence of that eDNA. Well, let's look at the shedding rates of various species, and that's what this paper covers, is like looking at carp's eDNA shedding rates. How often are they actually releasing eDNA that we could really use, right? There's another question of DNA transport distance, which is going to be really important to my modest contribution, which I'm heading towards in just a minute. Once the eDNA is released into the water, how far does it go? And what does that mean for us being able to capture it? And most importantly, what does it mean for us being able to indicate the presence of those types of species that I mentioned in the past slide or two? If we're getting DNA at some spot for eDNA and it's not where the species actually is or was, then there's no meaningful thing we can learn about these, the presence of these species from any sort of perspective if it's not accurate. So this was another methodological question that was important. And then this is, I think, another one, at least from David Pilliard in Idaho, along with Karen Goldberg. that looks at the persistence of eDNA present in streams through the addition and removal of salamander species. This is actually a really cool study where they had some good headwater streams with unilateral flow, where they actually took endemic salamander species that they weren't detecting in a stream. They knew they weren't there. And then they actually added salamanders to the stream and let them live there for a certain amount of time, and then detected the eDNA and then removed them again, and then looked for how long they could also detect the eDNA afterwards to learn a lot of things about shedding rates, transport distance, and persistence of eDNA in there. So this review paper that I mentioned from Thompson and Willerslev really led to a bunch of questions that people talked about in terms of methodological questions. These are kind of the big four buckets that I'm talking about here, but there's a bunch of others. Like, I remember reading so many papers about what sort of filter would be best depending on which matrix you're working in, as well as volumes of water or whatever matrix you're working in, as well as, do I use ethanol to sanitize the sampling equipment I'm working with, or do I use something else? And it turns out 10% bleach is probably the best thing. It's a really interesting study. There was a whole study in regards to whether DNA is actually fragmented enough from being in the autoclave. The answer is actually kind of no. So 10% bleach is the best way to go because it actually shears the eDNA into tiny fragments. With an autoclave, the DNA kind of sticks to the surfaces and is not really sheared. It's just kind of hanging on, at least statistically significant way. Not to say that, after autoclaving that, DNA isn't destroyed in some form, but that's not the intention of the autoclave. It's to destroy, microbial species, not to shear DNA. So it's a really interesting result there. So there's a bunch of other methodological questions that all came out at sort of the same time as far as that review from 2015 with Thompson and Willerslev is concerned. So it was a pretty cool time to be involved in environmental DNA when I was, because I was working on my research mainly from like 2012-ish, 13 to 2015. And it's right about the same time that these review papers came out, as well as these methodological papers, and sort of an explosion in interest of these questions, both from the methodological standpoint and whether or not we can actually use this as a monitoring tool. So my modest contribution was about this big old fish over here on the right. This is an Atlantic sturgeon. Let's see if I can remember. Accipenser oxyrhynchus. It's a specific species. I was working in this study area that you can see over here on the left. Richmond, Virginia is just up to the left of this screen here, or to the west. These, for those of you that are from the area, you probably recognize it, but this is Presque Isle National Wildlife Refuge, and this is the VCU Rice Center, if you're familiar. This is the mouth of the Appomattox, which is pretty, like, you should be aware of that because the Appomattox flows through Farmville, but the mouth of it is actually just east of Richmond. The Atlantic sturgeon, we were interested in tracking it with eDNA because it's a federally listed endangered species, I think still to this day, and we were interested in finding out where the suitable habitat was. So if we could indicate at least presence or absence in some form, maybe we could also help to identify where the suitable habitat was for their egg laying, because they're an anadromous species. An anadromous species means that a fish species lives most of its life in the ocean and migrates upstream to lay its eggs, and that's what Atlantic sturgeon do. The interesting thing is that during this time period, the Atlantic sturgeon were making what sort of people in popular culture would say the Great Return, which is they were extirpated, aka they did not live in the James River anymore, for up to 30 to 40 years. We believe it's because of water quality that they were not returning. So their listing as federally endangered is probably both accurate and not, and as far as it's possible that these Atlantic sturgeon were just going to a different place to breed while the water quality and habitat for egg laying was not of the quality that they needed while we were basically destroying the habitat of the James River. It's a good thing to see them return because it says something about our ability to increase the quality of the water in the James. And so we were, my modest contribution was trying to identify eDNA methods that would help us find Atlantic sturgeon without having to do what's happening here on the right. We had to gill net these species in order to get them and get genetics data and track them and understand how many are there. So because they were federally endangered, it would be really helpful for us to be able to identify where they are without having to touch them, and eDNA is a way to get there. And so I was brought in as a graduate student to develop methods to identify where Atlantic sturgeon are in the James River. Long story short, and this plot is from 10 years ago, so sorry for its relative clunkiness to what I would want to do now. We were able to identify a limit of detection based on biomass of juvenile sturgeon. So actually the way that we did this is we used mesochasm studies with juvenile sturgeon to understand sort of the shedding rates and how long their DNA persisted, because one of the big problems was that in the James River, regardless of timing of Atlantic sturgeon returning, there would be some level of biomass that was present because of juvenile sturgeon, and we knew that. So we would have to identify a level at which we could say that like this represents something that's real. So we estimated based on like egg laying rates and hatch rates and what the catchment has been for juvenile sturgeon, a very rough estimate of what we thought the numbers of juvenile sturgeon would be. And I have to really like pause here and say that this is the title of my master's thesis is proof of concept for environmental DNA tools for Atlantic sturgeon management. And it was just that, it's proof of concept. Like we really needed a lot more work to truly understand the juvenile sturgeon biomass. And I should say here that I'm borrowing methods from that Takahara paper that mentioned qPCR versus biomass. I was trying to approximate that so we can get a better idea of sort of the relative densities of Atlantic sturgeon, because just having presence-absence data wouldn't be enough to identify suitable habitat for egg laying, right? Like if you just had presence of maybe one, that wouldn't tell us whether they prefer the area that they're at, right? So we attempted to develop this limit of detection so we could understand what biomass meant, right? So that allowed us to detect biomasses that are, quote, real. In this case, they're in yellow. The standard curve is in these bluish black dots. So these are the standard curve for qPCR. That's why they vary a little bit. And then I plotted the qPCR results from one season of sampling for the various sites, of which there's a bunch of technical replicates in each of these. So it looks like there's one, but there's at least 3 in each of these to get the points that we have here to understand the biomass levels of Atlantic sturgeon. This was in the fall, I think, of 2013 or 14 that these data are from. So the yellow dots are above what we would consider the limit of detection based off our very rough estimation of the juvenile sturgeon biomass that's present. So these are real detections of eDNA of Atlantic sturgeon in the James River just east of Richmond from the fall of 2013 or 14. I can't remember anymore. Now, again, these are all super rough estimates because the ability for us to even know where the juvenile sturgeon are in terms of biomass also had to do with an estimate of how much the volume of water that's present in the stretch in my study reach, which is also an approximation because there actually hasn't been a good bathymetric study of that part of the James River for a long time. So it wasn't entirely accurate as far as both the biomass level and how much water is present, which is okay. I mean, this is just a start, but it's something to show. I mean, we showed proof of concept that this could work. This is a real DNA detection. But the problem is you've probably been looking at this the whole time. Let's talk about bathtubs. So we have a sloshing issue. You probably noticed at this point, too, that the James River is becoming far wider out here. And if you remember all my commentary on methodological questions of environmental DNA, I ran into a huge one here. To my knowledge at the time, this was one of the biggest rivers that anybody had worked in for environmental DNA. It's 7th order, it's tidal, and the salt wedge is pretty close to the rice center. It's brackish. So we have multiple problems going on here. The biggest problem is that we don't have unilateral flow of water. So it's difficult to say that if we get a hit at this site, B107, that it actually came from that site. There's just a lot of water. This is like a half-mile wide right here, maybe a little more. So our ability to say that even in that biomass model, I think we actually had a positive hit from B107. I think it was from Prisk Isle and the mouth of App. I think they were hanging out here, which would make sense because they have to osmoregulate, which is balancing salt concentrations in order to come up and lay their eggs further upstream in the James River. So it would make sense, but we know for sure that the water is not moving straight here. Like when there's a high tide coming in, the water backs up in this direction, which could mislead us. So not only do we have a very rough estimate of juvenile sturgeon presence, which established our limit of detection based off the volume of water, which is also inaccurate because it's not a good bathymetric study recently, but then the water itself is also traveling in multiple directions. And so we would need to understand all three of those things to have better detection, like more confidence in our eDNA detections. And I really only had time to summarize environmental DNA for Atlantic sturgeon for one season's worth of sampling. So this is really just a start here, but it is a modest contribution in terms of like the things that we realized is there's a lot more methodological questions that you need to customize for any sort of environmental DNA question you might have, which is right in line with all the literature commentary I was just talking about from the same time period regarding Takahara and the persistence of eDNA, adding and removing salamanders. We were running into similar problems here, and we needed to discover them. And frankly, showing that those problems exist is helpful. But there's a lot more work to be done as far as environmental DNA of Atlantic sturgeon is concerned near Richmond. Around the same time, so this is what's so interesting about me doing this at the same time, is that there's a couple of things that happened that also coalesced the methods a bit more. That's why the slides are in kind of the order here, is because this kind of happened before this. This paper came out, I think, in right around 2015, 2016 from Taberlow, which introduced a lexicon for us to be able to more linearly talk about what we're doing with environmental DNA, right? And this is partially because of the rise of next generation sequencing. Everything that I was talking about before was either presence-absence data with endpoint PCR or traditional PCR, if you will, or with quantitative PCR, which is what I was performing with the Atlantic sturgeon here with this sloshing issue. But what was happening in the background as like Illumina platforms were coming forward that were really helping us better elucidate the sequencing data that's available from any matrix via having small amplicons that were statistically assembled to a reference genome as opposed to shotgun sequencing, which shotgun sequencing was good for the time, but it was much more difficult. And as we developed this next generation sequencing technology, it meant that instead of us having to perform qPCR to understand quantity, we could do what we ended up calling eDNA metabarcoding, which would be looking at an assemblage of species based off sequencing results from sort of universal primers that are conserved. And that was starting around the same time that I was doing my work. And this paper was really nice to be able to show that you could look at different identification levels from species, genus, family, or order through different markers. And if you were doing DNA barcoding, that would be to a single specimen versus DNA metabarcoding, which would be multiple specimens in a bulk sample. And this would be from some sort of tissue sample. So it would be like multiple insects or something like that, all the way to what I was just talking about, which is eDNA metabarcoding, which would be taking an environmental sample from one of the matrices that we've been talking about the whole time: air, water, soil, feces, ice, you name it, and then understanding the assemblage of the ecological community that's present based off the metabarcoding effort. So this paper was really nice in terms of being able to coalesce our lexicon onto like what we were really doing. In my case, it would be for Atlantic sturgeon, it would be just eDNA. It wouldn't really be metabarcoding or just eDNA barcoding, because we were looking at a single species, right? So it'd be eDNA barcoding, not eDNA metabarcoding, because we're looking at a single specimen. But this language is really helpful because before that too, we talked about isolating eDNA from multiple matrices and all these methodological problems. But then as next generation sequencing came along, there was all these other layers added to it, because now we could look at a single species or multiple, talk about community assemblages or not, and then all those different methodological questions that are present. If you want to find something that's like kind of mind-numbing, I didn't include it here, but Thompson, same guy, was looking initially at freshwater species assemblages, but he eventually moved to seawater assemblages of fish communities off of the North Sea, like in the North Sea, like off of I think Holland and Holland, Denmark, that sort of area. And he started doing some of the first eDNA metabarcoding studies, but it was really like, It was really convoluted because there's a lot of individual primers as opposed to sort of like the universal primer sets that we have today. So being able to understand and be confident in what was present there was difficult because of the hydrologic problems I just mentioned with my own work. So yeah, I didn't include those there because they're kind of like hard to look at mentally with everything else. But anyway, Taberlow, this paper, as far as coalescing our language, was really helpful. So now to summarize up to this point, we had Willerslev, who had an idea about getting DNA from any sort of matrix. That was sort of established through a few folks in Spain, the United States, and Japan, a few others. And then there was a summary article that sort of coalesced these methods to some degree, which then led to an explosion of methodological questions, because that review left a lot of questions open as far as like, how do we get DNA from the environment and understand where species are? During this time, I had my own modest contribution with Atlantic sturgeon and indicating their presence in the James River based on their return from a previous extirpation. And then we reached a point where not only are the methods coalesced, but also now we are starting to get the language coalesced in the context of next generation sequencing, because eDNA metabarcoding is like really going to be the direction that everybody heads in. Now, for my own story with eDNA, at this point in 2015, we run out of money for Atlantic sturgeon work. It does not get funded through the NOAA grant that we put in. It's the only section that doesn't with regards to Atlantic sturgeon, which sends me off into a rabbit hole of UVA human genetics, because I wanted to learn some more genetics tools to take back to population genetics myself in species that were not human in the long run. I ended up spending like 9 years at UVA bettering my skills as far as that's concerned, which eventually leads me to Longwood University, which I'll talk about in a minute. And then, so what's nice is now we have some folks, Maya, releasing another review article, which also helps us better coalesce into the methods that you'll see. Like in general, this is like something that sort of is like a pathway to developing eDNA methods at this point that's pretty well articulated with regards to especially either just eDNA barcoding or eDNA metabarcoding at this point, which you can do qPCR or sequencing data on in next generation sequencing platforms from either Illumina or Oxford Nanopore devices. And I probably won't get into talking about the differences between Illumina and Oxford Nanopore technologies that much in this lecture, but just know that like this figure on the right is sort of a coalescing of all the methods that have been summarized with the current methodological problems, which mainly relate to still hydrological issues and positive or false positives and false negatives. But the usual algorithm for developing these things is kind of parallel to this at this point. So you're water sampling, you're capturing eDNA on filters, you're extracting DNA from the filters, you're co-amplifying and indexing with barcodes the different DNA samples that are present at various stations. You're doing some equal molar pooling, doing some high throughput parallel sequencing, and then using some sort of identifying tool to identify, based off some universal primers, what's present in the community. There's a little bit more to it than that, than she's not explaining here in this article. The usual algorithm before you even get to water sampling is here on the left, and that's kind of where I'm going to talk about as far as like where we're at with the work at Longwood University. The usual algorithm now, to say nothing of this, is to develop primers, troubleshoot those collection methods. That's really kind of A, as far as this is more like these methods that have coalesced on the right are more like if you've already troubleshot all of your issues and then you're actually monitoring the species present here. This is more like when you're developing the tools. So you develop primers, you troubleshoot the collection methods, you test those primers, you kind of do a lot of this stuff at the same time. You perform traditional eDNA assays with endpoint or qPCR, so in other words, traditional PCR or quantitative PCR, to sort of see if your primers are really working, and you can use those primers at that point to help you develop a metabarcode, and then you can test that metabarcode. So you're kind of like working in this and this all at the same time to sort of understand, okay, do my primers work for the species that we're interested in detecting? Are my collection methods kind of meeting the primers where they are? Like, are we actually getting enough DNA that these primers that we designed actually work? So then you test those primers to do that through traditional eDNA assays such as endpoint and qPCR, because it is costly to do high throughput parallel sequencing and develop those metabarcodes. So, but this is kind of the process that you end up in, right? So this is what we've kind of coalesced on through that history of environmental DNA that I've been talking about. So what are we doing here at Longwood University? The James River Association is really interested in freshwater mussel habitat in Virginia, and as far as we don't really know where that habitat is. We have an idea of what that habitat might be, but we don't know if it actually is where the mussels are. And so traditional field surveys of freshwater mussels is labor intensive and costly. Just imagine an entire watershed such as the Appomattox, and you have no idea where any of the mussels are. You'd have to hit every single tributary that you feel like is reasonable to see if mussels are there. And then of course the ones you don't hit, you're thinking, well, should we have gone there? I don't know, they could have been there. So that's why it's really useful for eDNA in this setting, is because we can work our way upstream in sort of a lattice network and go to various points there, get a water sample, and see if we actually detect a particular freshwater mussel species, and we can narrow the search space for our traditional field surveys such that it saves time and money, especially on the salary side of things, by identifying the areas that they should search, and then we can figure out from the trends from what we do detect and whether they actually get mussels there, what the suitable habitat is, so we can know better what we should protect. So that's what we're working on in my lab at Longwood, and we're collaborating with the James River Association and Virginia Tech to narrow the search space with eDNA for a bunch of different mussel species that are within the James River watershed. I should say that some of these environments are better understood in the James River itself, but the Appomattox is nearly unexplored as far as where the habitats are for freshwater mussels in this watershed. So if we go back and look at what the algorithm is, like I showed you a couple slides ago, the bolded points is kind of where we're at right now with me and an undergraduate student who's helping me with this. We are currently developing primers, we're troubleshooting collection methods, we're testing those primers, and we're going to perform some traditional eDNA assays once we test those primers. I should say, for these testing of primers that I've been talking about, the way that you test them typically is by having some positive control tissues. So you actually kind of go back a few steps in terms of the lexicon, and instead of getting DNA from the environment, from the matrix that you're interested in, you actually get a tissue sample. In our case, it'll be like a swab from a mussel, and we'll isolate DNA from that swab or from the tissue, and then use primers we think should work to those species directly in terms of a DNA barcode. And we see if those primers work before we develop any sort of meta barcode, right? Because we can use, if they're all to the same gene, those primers to develop a barcode and then be able to meta barcode from the water itself. So we're sort of in the barcoding environment right now while we decide where we're going to water sample and get those water samples and see if these primers actually work with positive control tissues. And then we'll see with endpoint PCR whether they actually do work. And then we'll take the time to invest in developing a meta barcode to see if we can get that to work with Oxford nanopore sequencing technologies. Just to show you here, we've got a table of design primers to various species that could be or should be in the Appomattox, but we don't really know if they're there. Let's see, one of these, which I'm trying to remember, actually, it's funny that I've never really been a mussel sort of guy, so I'm not as good with identifying the genus and species names of these, but there's a few threatened and endangered species in this list as well as far as primers are concerned, which are really important. The James spiny mussel is what I'm thinking of as far as its distribution. We have no idea where it is or if it's in the Appomattox for that matter, and that would really give some more credence to protecting that habitat because it's a federally listed species. But anyway, the primer list is here for the primers we're going to test, and we have some positive control tissues for a bunch of these different species in general to be able to see if these primers work at all. And then we're working on getting some water samples as we speak while we test these primers to see if we can do any traditional eDNA as far as looking at it with endpoint. It'll be endpoint PCR, at least for now, to understand some presence-absence data to see if this works at all. And then speaking of it working at all, we're considering these sites, which were previously used by Dr. Leach for some water sampling in the past. We're going to look to see if these are logistically possible for us to get to and then get out and get as many samples, water samples, as we can. The nice thing about sampling for eDNA is it has a 100% catch rate, and as far as you can get water no matter what, as long as there's no dangerous conditions. So all we need to do is go out and make sure that our bottles are 10% bleached to get rid of the DNA, and then that was there, and then we can go out and collect a liter of water for the sample and filter it, and then we can just catalog that while we test our primers before we go to doing a traditional eDNA test with endpoint PCR. So these are the sites that we're considering getting samples from to look for freshwater mussel presence. And that same undergraduate student, Nick Duhlman, is going to be doing some of this as part of his senior thesis, which is really cool. And then the basic sort of methodological output that we're working on here is we've got these primers designed in primer blast, and then we're going to do some DNA extraction from positive control tissue measured with the nanodrop. We're going to do some water collection at the same time in the background to catalog a bunch of filters. So once we have our primers tested, we can do the traditional eDNA assay. And then we'll look for whether these primers worked in some gel electrophoresis. And then we'll filter these water samples and then extract DNA from those water samples to see if the primers that we tested will give us any idea of presence or absence of freshwater mussels there. And if it does, then we'll work towards developing a metabarcode so we can scale this up and look at all the freshwater mussel species present at one time to understand who's there and how much from a bunch of different sites within the Appomattox, and then hopefully the rest of the James River watershed. So that's kind of a summary of these eDNA methods and where they come from. And to summarize one more time, they really started in 1999 with a guy, Eske Willerslev, who was kind of a naturalist in a way that we sort of thought of scientists maybe back in like the 19th century, where he was like, I have a really tough question I want to answer, and I have to go out into the world to answer it. And he just got an ice core and then started sequencing what he could from that ice core to understand the microbial community. And that really led to a lot of other questions with both ancient DNA and environmental DNA. And that led us to be able to, start to assay from various matrices, mostly water, frogs and salamanders and fish of various sorts that were rare and indicator species, endangered, or invasive species. And then we had some sort of coalescing of methods through various reviews that articulated the questions that were relevant, such as, how long does DNA persist in the matrix that you're working in? How much is shed off the species that you're interested in? And how can we better coalesce on methods so that when we publish our eDNA articles, we can actually understand what it is that we did and whether it's relevant to someone else and be able to reproduce those results. That was one of the big reasons why all these methodological questions were out there, because people found it difficult to reproduce those results. And I think we still have a lot of problems with that today as far as reproduction is concerned. There's only, I think, like 2 or 3 government-backed monitoring programs of eDNA in the world right now, just because it's just really hard to reproduce the results. We just haven't landed on methods that are linear and reproducible in a way that things like governments would rely on as of yet, but it could be a tool. And so that's why I'm interested in it. I think it's a really interesting interaction of molecular methods versus the ecological world, especially given that we can't actually see the species. But I mean, long term, it'd be nice with these metabarcodes to start to be able to get down to the individual level of, like individuals in a species, because then we could be doing like meta population genetics, where we're starting to identify individuals and then looking at genetic diversity within those communities, to say nothing of their presence and biomass there. I mean, I think that's where the long-term future is, but we'll just have to see, and we have to take these little baby steps, like seeing if freshwater mussels are in the Appomattox at all. So that's my kind of story about what environmental DNA is. If you want to contact me, I should have put it in a slide, but you can write this down. I'll say it slowly. My email is Hinkle, HINKLEJ, @longwood.edu, or you can come see me in my office in room 202 to ask me more questions about environmental DNA. And, shameless plug, I've got 2 students that work on my research currently. One actually doesn't work on environmental DNA. They work on an entirely other story, which I'll have to talk to you about later. But I'm looking for a student maybe to come in and learn from Nick while he performs his senior thesis, so you can take the torch and continue our methods, maybe develop this metabarcode actually. And if you're sort of a sophomore, which maybe in 427 you're not a sophomore in that class, but if you are, if you know someone who's interested in doing research who is a sophomore, tell them to come by my office in 202 or send me an email. And I'm happy to answer any questions you have about environmental DNA if it really sparks your interest. And I hope you enjoyed this lecture, and I hope that Kenya takes you through some pretty interesting papers. Thanks for your time, and I hope to see you in a class sometime.